Recently, OpenAI announced their current progress in accelerating life sciences research. In particular, they describe how they've designed proteins that are 50 times more effective than nature's own design. This is through a collaboration with the longevity company RetroBio. Today, I speak with Rico Meinel, who leads the applied AI team at Retro Biosciences, where he focuses on engineering proteins for enhanced cellular reprogramming and rejuvenation. In this discussion, we'll go into detail about what they achieved and how they did it. I hope you enjoy our conversation. So, if you go into Retro's website, you'll see that the main headline is that they're planning to add 10 years to human life. So, I guess I'm curious to hear your perspectives on Retro as the company and what sort of attracted you to join there as head of the AI research.
Yeah, so I think what attracted me to join Retro was really that I think in order for us to like make meaningful advances to actually tackling aging, we really need like a step change in how we approach it. I don't think, I guess I'm not the opinion that we currently already have working aging drugs and we only need like a break on the regulatory side and then we would tackle aging. I think we don't actually have anything that really works. So, you need actual vehicles to develop that. I think Retro is very well positioned to be such a vehicle and particularly because it takes a very strong like engineering approach to um, making medicines, and that's what really attracted me to the company. Um, I think we're well on track to do that.
Yeah, it sounds like it. And I mean, you keep using the words engineering. Now, maybe my viewers are not so familiar with Retro as a company. So, this is very ambitious what they're claiming. How exactly is Retro trying to achieve this?
Right, so right now we have these sort of two pillars of how we're going about this. One of them is replacement of like more micro replacement of certain cell types, and then rejuvenation, which is generally what we mean by that is in vivo tissue rejuvenation. Um, on the replacement side, we have two programs. One is a iPSC, so like this pluripotent stem cell derived hematopoietic stem cell. So it's like the cell that makes all your blood and immune cells. Um, that's one of our clinical programs. The other one is a iPSC-derived microglia cell for neurodegenerative conditions. And on the rejuvenation side, we have a small molecule for it's essentially an autophagy booster that we're using to treat Alzheimer's, and then a tissue rejuvenation program, which is more on the research side, but it basically looks at like how can we take factors like the Yamanaka factors or other sort of transcription factors and use them to rejuvenate tissues in an aged person in vivo, in of course like a manner that's safe. And yeah, those are I think the two main pillars that Retro is working on right now. And my team, because I'm leading the Applied AI team, which is sort of collaborating with all of these programs, and we also have like research efforts ourselves as well.
Oh, cool. And so I think this project you mentioned regarding the Yamanaka factors is kind of really why I brought you on today, because recently in the news, it was announced that this collaboration between OpenAI and Retro has achieved a lot of success in sort of enhancing this cellular reprogramming process. But before we get into the details of how exactly you did this, how did this collaboration begin in the first place?
Yeah, so I think I've been passionate about the idea of using AI to advance biology for a long time. I think biology is obviously very complex, perhaps too complex for humans to ever really truly design from first principles. Whereas we now have these models that are able to eat vast amounts of data and extract meaningful patterns and give meaningful outputs. Um, and I was sort of curious how we could apply such models to kind of the challenges that we were facing of like if you, for example, take the Yamanaka factors, how can you make them both more efficient and like more safe? How can you essentially extend the meaningful therapeutic window to be able to apply them in the clinic? Um, and from that, I sort of thought like, okay, or I guess like when we conceptualize this project, like we have these models like GPT that are able to like model all the text on the internet, and they are, you know, asymptotically
Hmm.
approaching, perhaps superseding human intelligence. And then you have these sort of like protein and DNA language models that have basically ingested the whole corpus of protein sequence and DNA sequence genomic data on the internet. And they're really good at extracting patterns there. What we're sort of like missing is like a bridge in between where, because GPT is not necessarily very good at protein design, and these models that only know how to speak amino acid sequences, you can't ask them any questions about like previous literature. So you're curious if we can do something in that space, and then obviously it would be nice to like test these things at a larger scale. And so we ended up partnering with OpenAI, who is of course very good at scaling these models to the kind of sizes where you would expect meaningful improvements. And then we essentially applied to this problem that we were facing ourselves internally, like I mentioned, to increase the therapeutic window of engineering the Yamanaka factors. And that's how we went about it.
I see. And so, yeah, how long ago did this project start? Just the past few years, right?
We started collaborating in I think April last year.
I see. Yeah, I guess, I mean, it's crazy how, I mean, even the GPT models keep on enhancing and getting better. And it's, yeah, I mean, we'll get to the achievements soon, but yeah, I think it's incredible within that time window how quickly you can make progress using just AI. But yeah, you mentioned this phrase protein design, and essentially what you've been doing, from my understanding, is trying to take the current Yamanaka factors, which of course there's the four Yamanaka factors that take fibroblasts or differentiated cells back to that pluripotent state, and you use something called protein design to make them better. But I think I'm curious to hear, to my viewers, to help them understand, what exactly do we mean by protein design or protein engineering?
Yeah. I think it's to me is the art and or science of I guess science of altering a protein's sequence or structure to achieve a certain function. Um, and I think uh, I guess I'm saying partly art because like the sort of like sequence space that we can optimize is like basically like not tractable because for a single protein, if you only take like three amino acid sites and you want to basically swap it to every other amino acid, you already have like 8,000 different sequences that you could try there. And now we are able to print DNA at a very large scale and we can test these things at a very large scale in the lab. I guess the larger you go, the more noisy your assay becomes. Um, but yeah, so there you're really like facing some limitations. So you cannot exhaustively test everything. So I think you have really great protein scientists that are able to, look at a protein sequence, look at a protein structure, figure out which domains are like would potentially be able to like if we manipulate them, we can get the function that we desire. And our yeah, so like but I mean, that begs the question, like, is there a better approach? And our approach to that was essentially that if we have a model that already has seen all the proteins, all at least the very broad space of mammalian proteins, like pretty exhaustively covered, I think for like, the whole space of bacterial proteins in the world, we're maybe not quite there yet, having that all mapped out. For mammals, we're I think pretty saturated. Um, and then seeing if such a model could help us accelerate this process of protein design to get the kinds of like functions that we're interested in.
Yeah, I think that's a very good explanation. Yeah, I think the way I see it is we have the original, like, I guess in this case, human protein sequence. And if we want to, in this case, with the reprogramming, make it more efficient than what's currently available, can we change this protein sequence in a particular way to enhance that efficiency? And of course, because we can extend the length of the protein, completely swap parts out, change amino acids, as you say, it's like an infinite thing. There's too many things, and I can't logically decide which may be best, which is why I guess AI is incredible with helping that process. So yeah, so that actually brings us on to what exactly you were doing regarding with using AI or this GPT-4B, right? The new tool, and there's four Yamanaka factors. Yet I believe you focus mainly on just SOX2 and KLF4. And so I guess could you explain a bit about your rationale behind even choosing just two of these factors to try and improve?
Yeah, so we had a sort of like literature proof of concept for I guess like okay to take a step back. I guess actually I think like maybe before this project started, at some point we sort of like mapped out internally of like what would be the so like the ultimate state of being able to do protein engineering, and it was really this idea of like synthetic transcription factors, where you really, I mean, I guess in that case, almost like de novo design a transcription factor that upregulates certain gene programs to be able to take you a cell state from one to another. Um, so like I think in the long run, and I'm not suggesting that our project is like exactly that already, but I think it's like a step on the way to go there. Um, but Yeah, so we had this literature precedence that I think there's a really great lab by this guy, Ralph Yao, who showed over many years that basically engineering a SOX protein to become like better at interacting with OCT4 during the reprogramming process and make that process overall more efficient. And we were sort of inspired by that to see like, okay, cool. If we take this work and we sort of like approach it from scratch with like having human scientists design variants of SOX2, how well sort of like are we able to like approach those kinds of results, and then also having the model do that. Um, and I think we did well, not quite as well as what they achieved with their super soxes. Um, and then we had sort of like our model add in the model designed sock sequence and the model designed KLF sequence, and then our sequences basically jumped up a crazy amount in efficiency, and we basically got this like almost 50x improvement in reprogramming efficiency. And I mean, at that point, you could of course continue with like now going to awk, going to make. I think make is probably a bit less relevant, for example, because when people do in vivo reprogramming, they mostly actually remove make because it's an oncogene, so you kind of like really don't want to have it in there. Ideally, you can get the same kind of efficiency without CMake. Um, I guess yeah, we could have, maybe we are internally still working on October, but I think the results are already like I mean surprising, and then as we validate them, also good enough to sort of like get something out there.
Yeah, I mean, the results that came in the announcement the other week are incredible. The way in which you've massively enhanced the efficiency just by improving the two separate SOX2 and KLR4, I guess retro SOX, retro KLR4. But yeah, I think what I'm interested in is trying to understand like how exactly was this GPT model able to achieve these results? And so I know that you mentioned already that the protein sequence itself is like its own language, and that we can give the model the human sequence and all of the other mammalian sequences, but is that all the information that you need to give it, or how does it decide, oh, this is the best design to make?
Yeah, theoretically, our model had a bunch of different ways of prompting it, and I think really sort of like as we evolved in the protein models, the initial models were sort of like sequence only. We had like individual sequences with these separator tokens in between to basically tell the model that here there's a boundary, don't like look across the to the other protein. So it was really like made to design individual proteins. Same as AlphaFold, right? The initial version was basically like you give it a protein sequence, it gives you a structure. More and more, we started adding like these protein interactions. And then some people have worked on integrating basically evolutionary context via multiple sequence alignment, where you show the protein not just one protein sequence, but many different sequences across evolution, across different species. Um, so, um, so essentially, we wanted to enable the model to use different modalities of information. So of course, it had text already from the initial GPT-4o model because it's initialized from a smaller version of GPT-4o. We then gave it this like tokenized structure data, so the model is able to understand protein structure. It's able to fold protein sequences. Um, then it can look at protein interactions as well, and we also gave it the ability to look at this like evolutionary context, all that in an autoregressive, so basically a model that just predicts one token at a time, um, yeah, autoregressively. Um, we ended up finding that for the Yamanaka factors, the structure information wasn't that useful because those proteins, as you probably saw in the blog post, are like basically like 70 or
Mm,
80%
yeah.
just disordered, so they don't have like a stable tertiary structure. They only form one when they interact with other proteins. Um, we don't have that kind of data. So it would be hard for the model to actually understand that. So we actually did really well by just like looking at the protein sequences. And of course, you have four factors. So, like there's interactions between them. So you can show to the model what factors you want to design, and it will basically fill in the whole protein sequence. And then maybe it helps to show different protein sequences across different species because um, the four Yamanaka factors are very strongly evolutionarily conserved. I think like SOGs even all the way down to like microbe level. So there's a lot of evolutionary context that you can take into account to basically see which domains of your protein do you want to potentially try changing, and which ones are so conserved that probably changing them will just break the function of the protein. And the cool thing, actually, I'm thinking of it not so much as a challenge, but as an opportunity because I think now we're becoming really good at designing and optimizing proteins with a stable tertiary structure. People with disordered proteins, people kind of like throw their hands up and it's like, oh, we don't understand those guys, we don't know like what to do with them. But actually, I think there's potentially a code to be learned based on like the sequence of those proteins and how that relates to function. And potentially our model was able to pick up on that, and that's why I was able to succeed.
Yeah, I think it's incredible that it can achieve it. And I suppose, yeah, I guess when my very more limited, I guess, interaction with like ChatGPT is, I ask my problem and it answers it relatively fast, maybe no more than a few minutes. But once you've given all this information into the GPT-4B model, like how long does it actually take to sort of process all this information and come out with its like short list of proteins or potential proteins? And was this like an iterative process that you took the first few examples, maybe they weren't so good, you tested them and gave it back? Could you sort of elucidate a bit on that process?
Yeah, so you can, I guess the output is basically instantaneous. Like, you give nowadays you put in your prompt in the ChatGPT, and it will maybe take like a couple of seconds to, depending on how hard it thinks about it, it will maybe take a bit longer, maybe like 20 seconds or something. But then I think like we generate, of course, a bunch of sequences, and then we had some like filters that we applied afterwards to like sort out the sequences that were like obviously bad, and have like some heuristics for like um maybe here it broke like a core domain that we didn't want to break, etc. Um, and filtered it down to also like a because I mean the model can give you infinite amount of sequences, but maybe you only want to test like a couple hundred, if even that, like in the lab. So we generated a bunch. Also, like try to like increase the diversity of the space so we can test like a diverse set of hypotheses in the lab. And then I think for SOX it was like a couple hundred. For KLF it was I think only 50 in the end that we took and basically validated in the lab. And I mean, I guess luckily we only had to run one iteration. So it was for both Fox and KLF, we did one screen, took the best sequences, and that's sort of like what we ended up with.
I see.
Didn't have to, yeah.
But in that case, if it's that quick, and I know, guys, of course, the bottleneck is the sort of like screening and testing process. But if it's that quick, surely you could just sort of
The
feed it
wet
back
lab is the bottleneck.
in.
The wet lab is the
I
bottleneck.
know, it's true.
Always.
Always the bottleneck. But yeah, so
We're gonna get there.
I guess, yeah, on from that, yeah, I suppose, yeah, I should say it's the bottleneck. But another idea that I thought would be interesting is that, currently you just tell the model, I only want you to sort of give me a sequence for SOX2 or just for KLF4. Why not get
Yeah.
it to sort of try and improve all four of them at the same time? Like, I think that would be an interesting approach.
I like that idea. I think we could have done that. Uh, we just didn't.
Okay, I see. And so at the moment, you've got these incredible results, right, with SOX2 and KLF4. And actually, the other thing I want to mention is the other crazy thing with your model is that it didn't make like slight changes to the protein sequence, like as maybe as humans might tweak it slightly to look more like a different species, SOX2 or KLF4. Like instead, it made massive protein sequence changes. And so, yeah, I guess do you have any idea why it did that, and also how did it change the protein?
Yeah. It's actually an interesting, maybe like philosophical questions. Like maybe I guess in biology, things usually don't work. So many people are like quite conservative in their experiments, or at least like they are conservative. Something works, then you go wild, and then something doesn't work again. So you like scale back and are more conservative again. I guess the model doesn't have any of these prior, so maybe it just went wild and like making some crazy sequences and see how they work. I guess just like come up with some crazy hypotheses, just test them in a lab and like let them crash. And if it actually does work, you can you can always scale it back to something that's more manageable. Potentially maybe you want to reduce immunogenicity. Like maybe not all the sequence changes the model made are actually necessary, so you can do a bunch of follow-up work to then remove them. Um, but yeah, I guess so interestingly, we don't know why these sequences work so well. I mean, we sort of have like an internal suite of like predictions for which domains may be responsible for the increased efficiency. Uh, I think that's I guess you can get that, but to really know, you'd have to test them in the lab. And if your model made a bunch of sequences and you kind of like want to like test domain by domain, and you have a bunch of sequences, that's a lot of like screening. Whereas like, we're here to make better therapies for patients. So maybe instead of like spending a year on doing that, we try to actually just like make sure those factors are safe and they perform well across the metrics that we care about for the clinic, and we actually try to get them to help with our clinical protocols.
Yeah, exactly. I mean, yeah, I'm more academic scientist, so I think there's like many PhD projects that could stem from just these findings. I think it's fascinating, in particular because, yeah, of
I,
course,
so yeah, I do hope we're gonna have some sort of arrangement that we're able to release these sequences in a way that people can do experimentation on them. I think we had a lot of interest, and I like, don't get me wrong, I do think that those follow-up studies will be extremely interesting. It's just like, I guess, yeah, if you're assembling like a plane while trying not to crash as a startup, maybe you try to focus on like, the things that will immediately get you towards like making products.
yeah, no, yeah, I think it's a fair conclusion. I guess I'm always just very curious. I mean, because I mean, I think, as you say, it's not probably immediately the most important thing, but if it's making a protein change and that increases like the stability, I think the things like half life of your therapeutic is things to sort of perhaps
For sure.
consider, right? Because I guess, yeah, this actually brings me on to how you are thinking currently as a company to deploy this therapeutically. Because if it's just a temporary exposure to these retrosults, retroclear 4, then I think it's a much safer approach than if you, of course, you were trying to have extended expression of this very heavily changed protein. Because, sure, maybe it reprograms, but if it then starts behaving like you've added neo functions, like new functions, by adding these new modalities, I think that is something
Yeah.
that's worth considering.
For sure. I think safety is definitely paramount here. And I think given that our reprogramming is faster and more efficient, so like the first questions that we had were, okay, does it mess up the cell's genome? Like, cells can acquire mutations throughout this reprogramming process. Um, maybe like in vivo, they were proliferating not as slow. Suddenly they're being put in a dish and forced to, you're basically dropping like these OSKM bombs on them, and every, cells around them are dying. It's like a very stressful environment, and you're like really pushed really hard to become an iPSC. So like you may have afterwards, you may have genomic or chromosomal abnormalities if you really push that process to be faster and more efficient. So that was like one of the things that we really wanted to check. And it turns out the factors are, well, the same, if not more safe than sort of the canonical factors. So that was like a thing that was really important for us. Of course, like we'll end up having to check that for every single line that we will program in the clinic. But also, we are like right now, the initial use case for those factors is ex vivo reprogramming, for these autologous cell therapies that we're creating. So I think there you always have the ability to like make sure to check for like, certain mutations that shouldn't be there, etc. Like the health of the cells before you're putting them back into patients. So that gives you like a bit of a buffer on the safety side of things. But as I mentioned, we are also interested in like in vivo tissue rejuvenation, and there you're absolutely right that you would yeah, also want to take into account potential immunogenicity of your factor. So if your model like really rewrote this whole sequences, maybe now your immune system would start targeting it. But yeah, as I said, like I think the model is really good for like quickly screening a lot of hypotheses, but then you can always go back and like turn the sequence into something that's more like safe and like acceptable from a immunogenicity perspective.
Yeah, I think so. Yeah, I think I mean, as you said earlier that your first maybe idea was to create completely de novo novel proteins, which would be super cool. But even starting with the baseline human factor already sort of adds that hopefully additional safety. And of course, that's what the sort of next steps are, is to try and make sure these factors are safe. And I guess actually, yeah, regarding the next steps, so currently, you found your RetroSalx, RetroKLR4, and you're doing in vitro screening. Like, are you currently planning to do any sort of preclinical models, or what sort of stages is that at?
Yeah, I so I guess since we sort of like concluded that project and now give it into the hands of the teams that are attempting to bring this into a clinic. We are now, I guess, more focused on. So I think the second result that we showed in the blog post was those the I guess our portfolio of factors is not only able to increase the reprogramming efficiency, but also based on like this paper in 2016 by one of our advisors, Alejandro Ocampo, he showed that you can use the four Yamanaka factors, maybe use three in that paper, to basically reprogram in vivo and ameliorate multiple hallmarks of aging in mice, and also increase the lifespan of progeric mice. Um, and I think that's something that we're really excited about. Obviously, and have been working on for a while. And in our paper, in the blog post, we sort of showed that our factors are able to sort of like OSKM is able to reduce DNA damage when you stress your factors with a chemotherapy. When you stress your cells with a chemotherapy, OSKM is able to sort of like increase the DNA damage repair response to that, sort of like make the cells more resilient. And then when you apply our factors with like the like OGG, MYC, and then RetroSox, RetroKLF, you're sort of able to further increase the like rejuvenation abilities of the cells by like further increasing DNA damage repair. So that's actually the result I'm also really excited about for, potentially making like yeah, safer but more efficient in vivo rejuvenation therapies.
Yeah, that's very cool. And actually, one thing I've not asked yet is currently we've kind of described this success as sort of the complete reprogramming approach by going back to the iPSC cell state. But of course, in the longevity space, some other companies, and there's also a bit of interest around partial reprogramming where you just take like a very brief exposure or you use just OSK and sort of partially rejuvenate, but you don't fully reprogram. As Retro also considering, I mean, maybe your current RetroSox RetroKLF are too powerful. Maybe they only can do complete reprogramming. Could you sort of maybe make perhaps less efficient, tell it to make less efficient models that just sort of slightly rejuvenate?
Yeah, yeah. Oh, so sorry, I should have clarified. So in the last point that I was making about the DNA damage repair, so we do partial reprogramming there.
Oh, I
So
see.
instead of waiting like I don't know, like two weeks or something, we only expose the factors for I believe like 4 days or something. So the cells are not iPSCs at that point. Maybe they have like initial markers, but they're only partially reprogrammed. They're still very much fibroblasts. And you're and I think we've also seen that maybe the factors who are better at the DNA damage repair, um, as a proxy for rejuvenation, are not the most efficient reprogramming factors. Um, I guess some do overlap, but maybe suggesting that with that kind of design approach, we are able to decouple the ability to reprogram and rejuvenate on the sequence level.
I see. That's cool. And so currently you're doing more moving towards more preclinical models. But what is Retro's sort of timeline with this project? I guess the aim to get it to eventually human clinical trials.
As soon as possible.
Nice.
Um, I think year or two. Um,
Yeah, I
I
mean, it's
guess
already
as they
incredible.
say,
Yeah.
watch this space.
I mean, I think it's really
um,
worth emphasizing how quickly, like, I went to Retro and, like, saw the company when it was in sort of crates and boxes. Perhaps, I think it was only 2022, so only 3 years ago. And to see it from that to where it is now is already incredible. So I think the speed with which the company is progressing is very rapid.
oh yeah, I think it's important. Patients are waiting.
Exactly. I
And yeah, I mean, as I said, like I think I mean, I think myself, speaking for myself, but also like I think essentially everyone at Metro, like we really want to solve aging in our lifetimes, right? Um and like I think going for proxy indications, as we do right now, is really important to establish the safety of our approaches and therapies. Eventually, we do want to run trials for like extending like health span in humans, and those trials will take longer. Um, Maybe we'll even need some like different approaches there. So, um, yeah, I mean, I guess with aging and those kinds of studies in general, like time is not our friend. So, trying to like squeeze it as much as possible while not sacrificing an ounce of like quality and safety, I think is really the key to succeeding in that space.
Exactly. And I mean, as you said at the start, Retro doesn't just have this one Yamanaka program. There's a few different programs that are occurring. And I guess, yeah, and if you're allowed to speak about it, so far we've spoken about how you used AI to help with the Yamanaka factor protein design, but how else are you using AI in perhaps your other areas at Retro?
We I guess with this approach we sort of like specialize ourselves in design of proteins. So we had another program within the company where we redesigned like a receptor um protein to like essentially help it like that protein has effectively a kill switch at the membrane, where like using a small molecule you can tell like certain cells to commit apoptosis. You can design the protein so that it's no longer um like able to receive that sort of like kill switch message, and that would it basically enables something for one of our cell therapies. So we had other in-house proof of concepts where we designed other proteins other than Yamanaka factors, and I think maybe that got a little bit lost also in some of the initial press releases. Like this is a general model. This is not a model designed for the Yamanaka factors nor a model designed for longevity. We really build for like the general use case here, and then we sort of like showcase that this can work for um, our Yamanaka factors because that's like the most pressing sort of like problem that we were facing at Retro. And we thought it was a great proof of concept because nobody had shown that you can actually, with AI, you can make these proteins more efficient. As I mentioned, they're really hard targets, etc. But we do have, I guess, other projects that we're working on for redesigning other proteins.
Oh, nice. That's really exciting. And yeah, I think one of my other questions is, I've been, maybe, I suppose my career in science has not been as long as many people, but I feel like I've been in biology now for a long time that, I really can see the difference between how I did my research before and after these models all coming out. And I know that what I use is far smaller than what Retro's been using it for. For me, it's more just like, how does my experimental design look, or can you help me understand
Yeah,
this
it's a
concept?
great application.
Could you, yeah, I
Yeah.
mean, you've been in AI for a long time yourself, well, yeah, maybe similar time to me in biology, but how do you think that we can use AI in, besides protein design, like what are the applications in which we could be using AI in biology?
I think there's always like this window of what can we do right now with the data and the models that we have available, and then of course you have the ability to generate new data to sort of expand that window, and then like what is actually relevant. I guess like I think what's exciting is that we're getting better and better at understanding like molecular structures and sequences um, and genomic context, etc. Um, and then, but I guess like then the question is like, what are the real problems that are sort of like causing this emergence of like Eroom's law, where it becomes more and more expensive of to like make new drugs, and I guess like two-thirds of drugs fail in like phase two, phase three later trials, and most drugs don't like translate from like smaller organism in vivo models to humans. So if those are like the major bottlenecks of the field, maybe even if we have like perfect in silico design of like new antibodies, new binders, etc., maybe the field still won't advance that much. So but I think there's definitely like room for optimism because I think it's definitely great that we're making a lot of progress on these fronts, and like I think nobody will say no to like the perfect binders, perfect antibodies. I think it's just important that we sort of like align the objective of like the models that we're building with like the metrics that matter, which I guess is like how many drugs we get into patients that are safe and actually like cure, or I mean ideally in my ideal world like just prevent diseases by tackling the underlying aging process. um and I think I'm excited if the field moves more and more into that direction, like deciding what those metrics are and collecting the appropriate data and working on those problems.
I think they're really interesting ideas. Yeah, because I think from my perspective, there are some kind of obvious bottlenecks in biology, and one of, as you've mentioned, is going from like actual research to clinic, getting drugs free successfully. I guess from my academic side, it's more about like data reproducibility and data quality. And so, yeah, I think, in my
Which is
opinion,
also really important. Yeah.
exactly. Yeah, I think they really are the two major bottlenecks, and if we can use AI to improve that, that's great. But yeah,
I mean, I mean, I do think every sort of like data set has its own sort of like noise properties, and I guess the benefit that we've gotten like basically for free with the sequence data sets with all the amino acids and DNA sequences, well, I guess most of the amino acids come from the DNA sequences, but like they're just like there's clean data, they're like ground truth
Mm.
data. You don't have to do any data cleaning really. Like I guess like talking to some of the OpenAI folks, like a lot of the processes, like you scrape the internet, most of it is junk. You have to actually clean it and get the good parts, and that process is really important. For proteins, it doesn't really matter. Like you just get the whole data set, you know everything is clean. You can do some like clustering to remove duplicate sequences. Um, but for example, RNA-seq or single-cell RNA-seq data, quite frankly, most of the data is junk that we have out there, and there may actually not be that much signal other than like, we have three different, you know, blood cell types in here, but maybe the data is not high quality enough because it's from like a previous generation platform that your model would actually learn anything meaningful about cell state. So I do think that in that space, for example, like, and I've worked a lot in these models, like the data quality is holding back actual progress on the modeling side of things. So I mean, I do think you raise an important point here.
Yeah, absolutely. I think for me that is really important, and I hope that like many biologists can use AI, even if it's only in smaller cases, to actually enhance their experimental methods and sort of work together with the AI models.
Or even like, I guess in the case of DNA, it's also very different than proteins. I think like if you throw your genome into a whole model and like 70% of the genomic sequence are just like retrotransposons, um, and maybe your model would immediately learn to just discard them and not pay too much attention to them. But then I guess we haven't had yet like a really great proof of concept that these models trained on like all the DNA actually extract as meaningful patterns as the protein sequence models do. Maybe it's just a question of we need more data. Maybe the sort of like just raw objective function that you're throwing at the model just like predicts the next DNA token. Maybe DNA's information content is so low that it will not end up picking up meaningful signals like which transcription factors bind here, or like how does this promoter how does this UTR like affect the stability of my mRNA product or something. And we just like need like orthogonal sources of data to actually derive meaningful information from that. I mean, yeah, as you can maybe tell, is like data quality, what data we collect, how we throw it into the model is like an area where I'm really passionate about. And I guess I'm lucky to be in a situation where we think about this not from a perspective of like what are the data sources that we have, but also like how can we use them to answer those kinds of questions, like how do cells age? Maybe if you have all the transcriptomic data in the world, but about like a bunch of aged cells, maybe the transcriptomic data does not contain the information about how cells age and what you need to do to reverse the age of the cell. Um, so maybe the data modality isn't exactly paired to the questions you're trying to ask. And then even if you have the best model, you will not end up being able to translate your insights into like useful therapies.
Yeah, absolutely. And well, I'm just looking forward to the day that I can see AI better applied to my microscopy images, so I don't have to painfully get through those too. Yeah, honestly, I know there's already artifacts. Like I think as you go back, like why is it issues with microscopy? Of course, it also depends on your staining process, antibodies. So yeah, there's always multiple levels to it. But yeah, that would be
Mhm.
very very nice for my situation. Or proteomic data, but I guess yeah, we've spoken a lot about the sort of amazing things about using AI to help biology, but I do think it's important that we also think about the perhaps the misuse cases of this technology. I mean, I know you've worked in AI, but AI more broadly, there's some people working on like AI safety, and I wonder like how can we also use AI in a way that's responsible and it doesn't create public fear about sort of bioweapons, etc.
Yeah, I guess I'm not sure if I have like particularly hot takes here, other than maybe the like. I guess at this point we have like the models that are out there are already powerful enough that any like sufficiently motivated bad actor could already use them to design some pretty advanced bioweapons, maybe. Um, I guess it's sort of like a balance, right? Like I think for like maybe in the LLM case when like OpenAI initially released GPT-2 and it was like a research preview because we were worried that these models would um like be so powerful that they could come in the wrong hands. And now having like GPT-5, we may look back at GPT-2 and be like, well, I guess maybe we were a bit off there. Um, so I guess there's definitely a lot of like hype about AI right now. I think it's definitely important to ask like what's the actual utility people are deriving from it, and if there is utility, that's awesome. We should try to derive it instead of like fully suppressing the like small amount of or large amount of utility that people are deriving by like being overly focused on like some security details. Um, I guess maybe that's all I would have to say to that.
Yeah, I think I guess I mean you're already focused on your own projects. I guess I think just generally maybe there should be more just other initiatives out there to really sort of consider the biosecurity. I think I mean even before we had this technology, it's always been a concern that I think people have overlooked, and there's never been enough research or sort of focus around it.
For sure.
So yeah, I guess we've spoken a lot about what Retro is doing and with AI. How we've spoken a bit about the timelines, but where do you see Retro as the company like in the next like 5-10 years? Do you have like what would you really like to see happen?
I mean, I would like us to be in the clinic for sure. I think that's a milestone for every company. I guess every up and coming biotech startup. I mean, it's important to have gone through that. I also think, as the aging field, I think we need to come up with some sort of like metric showcase to show that this is actually possible. Um, I mean, I think we've seen it in mice, but for example, like some people argue that maybe Ozempic and the SGLT2 inhibitors are already like a first showcase for longevity drugs because, they're able to reduce all cause mortality and maybe the obese or diabetic population. And I guess there the showcase is really strong. Like maybe you don't even need statistics. Like your friend starts taking Ozempic and they suddenly lose like 15 kilos. So you're like, okay, wow, this works. What does that look like for aging? And I guess on the eventual like in the eventual trajectory, like you'll want to, like I really believe that like humans age like fairly linearly, like we're stable throughout most of our lives, and then I don't know, like starting at like 60, suddenly there's an exponential decay. And for some people it happens earlier, for some people it happens later, but it definitely seems to me like because of this exponential decay, maybe even if we like cure some of the age-related diseases, we're not going to do anything. We're not going to actually add that many years to health human lifespan. And Retro's explicit mission is to add 10 years. Um, so I think with this whole approach of like replacing and rejuvenating, I think we're really, in the long term, I want us to be able to really like prevent diseases from happening in the first place. Treating like an otherwise healthy population um, so they don't get those diseases rather than waiting until those systems have already broken down and it's really difficult to revert them to like a healthy state. So that's something that I really would want to see over the next 5 years.
Nice, yeah, and I think it's of course very ambitious what Retro set out to achieve, but based on the current announcement, I think you're already heading the right direction. I think of course there's probably still difficulties with how you can prove that you've added 10 years to human life, but I guess well, I'll just have to wait and find out how Retro pulls it off.
Yeah, there will be signs.
Awesome. Well, thank you so much, Roger, for coming on today and explaining to my viewers and to myself how exactly you've been able to make these amazing improvements in reprogramming efficiency. I think, I'm certain that, I'll hear more interesting and exciting news in the future.
Thank you so much for having me. I love your show. It's been great to be on it.
Thank you.